they play, based on their wrapping, portions that they create within the brain. Dural sinuses. What was a sinus? Something from part 1. What did it mean when you heard the term sinus? A cavity. An empty space is opening, this like indentation, something that was open. Okay? Dural. This is going to be the space, because we have all this type of structure that's present that gets to collect blood that has circulated around the brain and help it empty into our internal jugular veins, which are going to return to vessels that go back to the heart. The blood supply to the brain is tremendous. I mean, tremendous. We're going to have the dura fold inward. So if I'm looking at this model, this longitudinal fissure, the wrapping are going to be able to fold inward so that I get the separation of the two halves. That is going to be called falx cerebri. We got real inventive in labs like that. On the cerebellum, same thing, because it has to separate the cerebellum. That one, the falx cerebellum. So that you get that nice little division. So you get roughly down to that corpus callosum, where it gets really, really thick, and it's very difficult to get a cut through that area and separate a brain that you're dissecting. I mean, that is one tough area to get cut through. It's almost like you need a Dremel tool. Okay? But it's very tough to cut through it. These wrappings that we find associated with the brain, one of the things that we talked about is how we're going to find some fluid that's going to wash over the brain. We're going to have to make it, well, let me ask a question. I'm sorry, that late entrance totally disrupted and disrespected my class, and I lost my entire train of thought. We talked about late entry on the first day, which you were not here, but you were supposed to watch the videos, okay? And I've totally lost my train of thought. Please don't enter late again. So let's think about this for a second. Cerebrospinal fluid. Is that something that we continuously make, or is it something that we just have a certain amount of and that's it? How?
I'm not sure how, but it does.
Okay, so then it's not anything that we have just a given amount. It's something that we're going to constantly produce for the cerebrospinal fluid. So, when we're looking at this, and we're looking at the brain, have you ever wondered about that fluid that's circulating around the brain? What, like what? Like, what's it doing? It's there, okay? What does it do? What's its job? Nourish it. Do what? Well, let's kind of think about this for a second.
I guess provide like a certain layer of protection, also like keeping them at edges and everything, fill with fluid, make sure it's proper cushioning.
Okay.
Especially like the spinal cord, things like that.
Exactly. Because it's a good cushioning that's present. But now let's think about this for a second. Because remember, everything in this class, every topic we learn, we have to get it down to the cellular level. If we know that the cerebrospinal fluid is washing over, nourishment, okay? What does that mean that my neurons are doing? Think about this, okay? Neurons. We have billions of them. And you gonna disrespect me by talking this way? I want to hear what it's about. Don't do it. Now, the cerebrospinal fluid, the neurons, billions. They got to do everything. They are getting us to have higher order thinking. They're getting information, making sure that we have our breathing rate, our heart rate, blood pressure, chemistry, balance, muscle control. Do they take a break? No. All right? Well, now, once you get a little bit older, okay, they do. Just a little bit. The things just kind of like, they're busy. What? Are they duplicating? Are these cells that are duplicating? No. Okay? Their only job, make their product. Their product, which we've already talked about, because up here, you've got that great big huge nucleus, which means that that thing's busy, okay? Because that holds the DNA to make the protein, and if it makes the protein travels and gets released from those terminal ends into the synaptic cleft. Remember that? Okay? So the only job they're doing, make their product, which we know as a neurotransmitter. What are some examples of neurotransmitters? I know you've heard of them. Neurotransmitters. You hopefully learned about one in part 1 for skeletal muscle movement. No, well, now, dendrites are going to be an area that is going to be receiving stimulation. Do you remember what was released into that synaptic cleft? It's acetylcholine. That is a neurotransmitter. That is a product, i.e., a protein that had, because the only thing a cell can make is protein, okay, that had to be made to be released into the synaptic cleft. Other neurotransmitters that you've probably heard of, maybe on TV commercials, read in magazines. Listening? Serotonin. Okay. There are so many. All right, just some we're more familiar with. Okay. We're more familiar with them because there's medications that have been made that can affect their behavior. Is that true? So now, think into my cerebrospinal fluid. If the cell is making a product, it needs nutrients, which you guys said the cerebrospinal fluid is bringing in nutrients. If that cell is functioning, making the product, it is creating waste. Do you remember that for cell protein making stuff? Okay, from part 1. Can the waste remain in the cell? No. Just like any other area of the body, the waste cannot stay. Cerebrospinal fluid washes over the cell, picks up the waste. And because of the structure that we find, it will allow that waste to empty back into the bloodstream, and because of some of the other structure we have, we allow nutrients to move in. There is only one nutrient that the cells can use, the neuron. Only one. We mentioned it. Does anybody remember? Marley should let somebody else have a chance. Anybody remember? Glucose. That's it. It can't use fat. It can't use protein. The only nutrient it can use is glucose. And your body's going to do everything it can to provide the brain with glucose. The way that we're going to create the cerebrospinal fluid, there are going to be areas in the brain that are going to be ventricles. Those ventricles contain specialized cells that will give rise to the fluid. There's a total, and when we look at these ventricles, we've got 2 laterally. That means you open it up, each side of the brain. 1, 2. All right? We're going to have 1 and 2 connect to the 3rd ventricle in a median space in the middle. The 3rd one going to connect to the 4th, and that's going to be the cerebral aqueduct, because this fluid, when you hear aqueduct, what does that mean? Running water, water passage, movement. Okay? We're going to have to get it to the spinal cord. So it's going to move through the spinal cord tissue just like it does the brain. And the central canal is going to help to do that. These ventricles, the specialized cells, they're termed the choroid plexus. We got a spongy mass of capillaries. Capillaries. We haven't discussed blood vessels yet.